I'm currently working with gene editing in potato. So potato is a very important crop. I think it's the third most consumed crop in the world. There is a lot of potential in promoting gene editing in potato, but because of its biological traits, it's a bit complicated. And it's mostly propagated asexually, what makes breeding a little bit difficult. And it has a potential for gene editing as many other crops, either to improve yield or tweak some characteristics of the plants. At the moment, I work with establishing a transformation protocol. And what I do is basically transform protoplasts transiently. So what's what we call DNA-free transformation. So we basically promote the gene addition in the protoplast and then put these protoplasts that are basically cells that we extract from potato leaves in medium that will promote their division and regeneration in another plant. So with that, we do the precision breeding. That's basically precisely gene editing the genes that we want without making the plants transgenic. So that has a huge potential, and especially now with the Precision Breeding Act. The protocol I'm working with now, it's challenging itself. If you think about, we take a leaf of a potato, we digest this leaf to isolate the cells, deliver the machinery that will promote the gene editing in those cells, and then we'll regenerate a single cell into a whole plant. So it's a very delicate protocol, and we have to pay a lot of attention to details. So minimal variations, that they make a huge impact and a huge difference. When we talk about gene editing, especially with CRISPR, right, the sky is the limit. So we can tweak genes to make plants resistant to drought, resistant to herbicides, resistant to pests in a world that is now changing at a very fast pace with areas that nowadays are arable and probably in the near future will become very hot and dry. We need to tweak plants to adapt to these environments. So working with gene editing, especially with CRISPR, in such an important crop as potato, it has a huge impact, especially in food security. We're still in a very early stage. So we are establishing the protocol first. It's such a challenging protocol that every little things, that better temperature, the light intensity, these little findings are already very important for establishing the protocol, and then we can transform potato and do gene editing in potato in a faster and larger scale. It's very exciting, and it has a huge potential, both in an agronomical potential and also for research. I think the possibility and the ability to actually improve conditions for farmers and also for the consumers. Experiment anything that you can. I mean, crop science is a very wide field, and there are so many things that are important and are interesting to be working with. And I think at a young age, as an undergrad, it's nice to experiment everything. I think here we have a very special combination of conditions. That is, we work collaborating both with commercial and stakeholders and also with research centers. So I really like that balance, and and you see the differences and similarities between them, and I think that's very, very interesting.